Gianna Cannestro, who she's from Florida Atlantic University, and she tells me that the paper on this work that she's presenting is being written as we speak. And she's going to talk to us also about tactile perception and human neuromarkers in tactile perception. Gianna.
Okay, so let me go ahead and share that. Okay, so we're going to be looking at the human neuromarkers of tactile perception, kind of discussing some issues and things we need to work on in that area. So of course, we want to restore some sense of touch in those who are limb absent, whether traumatic or congenital. We need to develop these tools for unobtrusive study to implement them into a sort of bimodal interface, kind of like Catherine mentioned, having from prosthetic to brain and the other way around. We do need to look at the user's brain for these responses and trying to make sure that we're keeping the tactile event as uninterrupted as possible to keep it as natural as possible in terms of the recording of the sensory input there. So the sense of touch is, I'm sure as the panel is familiar, one of the least well understood senses here. It has a very complex pathway. While we would ideally have a figure like this look like this lovely freeway, it often looks more like this with converging and diverging pathways here. So for this particular talk, we are looking at both more simple and complex sensory qualities here. So just a quick overview of some apparatuses that are being used. There's a lot, and I'm sure quite a few of us are familiar with a lot of mechanical stimulation that are being used that are custom or purchased specifically for that purpose, that then make it difficult to reproduce those experiments as results and get more of a universal response there, more specific. So for oscillations, we have the theta, we have some sigma, mu, or alpha, which the naming of that in itself is also an issue in the literature as well. Beta, and from the literature review that I'm currently doing, there's a suggestion that there's potentially a difference between beta that can be entrained and other beta, gamma as well, and then the separate sigma kappa burst, the ultra fast there. And this is just from the overview that has been done, just to see what responds to what, where we see these oscillations presenting themselves. Some of them, like mu that we were discussing earlier, tends to present, in the suppression rebound and is often coupled with some beta. Gamma is a little bit more picky, sometimes with pain and things like that. And then, of course, there is the fun confounds and covariates that like to throw in our little, throws in a monkey wrench there. So these are just some of the things we do see for these oscillations for each specific frequency band. We do see that different covariates and confounds here are affecting them differently. So theta, mu, based on attention, suppression, but some as the beta entrainment there as well don't necessarily have any identified, the same with the sigma and kappa, the ultra fast, that aren't as well studied. And even affect, like Luke was mentioning, what may be painful for one person may not for another. So these qualities of what is pleasurable, what is painful can also affect all of these, the expression of these oscillations that we're looking at as well. So there's a lot to do. We've got a fun little checklist here. So we do have to work to kind of level the playing field here and to understand each of these oscillations and how they correlate to their confounds and covariates as well, with, again, specific attention to the naming, the recording methods, all of the nitty-gritty details in there. And then also the development of specific stimulation techniques, specifically hybrid interfacing using, pressure and vibration, or, any combination of simple tactile events. But it also needs to be very precise, both spatially, time-wise, in the recording and application too. And of course, this has an application then in the rehabilitation and teleoperation, and the development of prostheses that work bimodally as well. So any fun, seems like we've got maybe some fun questions.
Okay, thank you very much, Gianna. That's you really highlighted a real issue, nomenclature, and this is why I'm a best practice aficionado. And we've got together guidelines, Covitus guidelines, which are available on the Open Science Framework, and that was the first article that I put there about nomenclature and frequency bands. There is a and then I've put in a perspective article to Nature Neuroscience where we discuss that. Very, very important, particularly for people trying to do meta-analyses and trying to understand the literature like you're trying to do. People are not using guidelines that the International Federation for Clinical Neurophysiology advocate. And this is stuff that we've also tried to incorporate in our Covitus guidelines for OHBM. Very, very important. And thank you for mentioning that. It's a real problem, and particularly when we get into these sensorimotor rhythms with, we're in the mu region and they have two components. One is sort of in that 10 hertz region, the other vicinity, the other one's at about the 20 hertz region. People name them alpha and beta. And that confuses people in literature because their reactivity is very different to the posterior rhythm that we call alpha. So that's a real challenge and a real problem. So thank you for highlighting that. And I'm sorry, I'm going to get off my soapbox now. Do the other speakers have questions for Gianna?
Sure, yeah. Gianna, thanks. That was really interesting. You mentioned the need to develop a stimulation apparatus, and one of the main points being you wanted to have good spatial, temporal, and functional precision. And you highlighted a lot of different types of stimulation, like mechanical, right? And then maybe there's electrical. If you had to guess, or what you're thinking on which method or modality might be best in some regards, or do you think some stimulation approaches might be good at one thing but not others, and is there a way of working around that so that you get the best of both worlds? And sorry, that's a really hard question because obviously it's something everybody's trying to find the answer to, and it seems like you thought a lot about this, so I'm just curious what your thoughts and opinions are on what type of modality you think might be most fruitful or not.
I do think that the development and utilization of stimulation apparatus that can potentially apply different sensory qualities, like the one that I mentioned. One of the developments in my lab is a vibrotactile, but also pressure applying. It's an armband. That's something that the reliability of. You can have one or the other. You can measure the specific application time and duration and things like that, as well as choosing to do multiple or just alternating or just one. I think a lot of the temporal precision is not only in the specific stimulation application, but also on the recording side as well. I think there needs to be a lot of development in the creation of these stimulation methods, but it also needs to become more of a across the board acceptance and utilization of these techniques, because even if they're there, if we're still using the things that we've been using forever, it doesn't really help us get that nice even comparison across the board.
Sure, and it may be interesting to think about, if you say you have an apparatus like that, it'd be cool to think about adding in additional modalities like temperature, right? Like imagine you're able to put in like a little thermal heater or cooler in addition to your vibration and your pressure, right?
Yeah, for sure. I think the most that we can get into one while still maintaining the integrity, the precision of that would be ideal.
Absolutely.
That's very, very. I mean, lots of new developments. Let me ask you a question, you guys, about EEG. Of course, is really developing now with these portable EEG systems, and you want to talk a little bit more about bringing this stuff out of the lab and home to the person's home, and how you see that developing. And this is a question to any of you.
Yeah, maybe I'll start. I guess anytime I see anything that has like the translation or mobility aspect of it, I get very, very excited. And then seeing how well it works is maybe a different story. But I think something like an EEG cap that you can wear around and perform activities of daily living unencumbered, that's pretty exciting, because maybe like the work Catherine and I were showing earlier, everything is very structured and in the lab, but what happens if it's not structured? Do we see the same effects, and is that a good thing or a bad thing? So it makes me excited, and then it's one of those things that makes me very excited.
Gianna, Catherine, you want to comment about that?
I think this is actually my third question, that what role does EEG play in this context? And I think mostly what we see right now is that it kind of looks at the brain level activity and serve as a validation of experimental interventions. And I think this is, I think it's a question that I have and I ask myself all the time, and it's just like, why do we use EEG, and what can we, is there a value to bringing it out of the lab? Because we see the reality with these experiments is it is a very long preparation time. But if we move to, let's say, fewer channels or the dry electrodes, do we still get similar observations that we see here? That's a question.
Mm-hmm. Gianna, you want to weigh in on it?
I think, Catherine and Luke both had really good points. I think the development of EEG caps or apparatuses that you can use and take with you, definitely, I think they would have its place for sure. Again, my concern, similar to what Luke was saying, is maintaining the integrity of the quality that we're receiving there. But I think if, all of these, every little step, every little blip is always, moving forward still. And so its eventual development of a quality and reliable portable EEG is something that I would really look forward to, especially for that precision, that temporal precision.
Yeah, we've been playing a little bit with portable EEG. Not that it's something that I particularly want to develop in the lab, but we played a little bit with it. And it's remarkable the kinds of things that can happen when you take it out into the real world. Electronic scooters, if they have the same Bluetooth frequency that your portable EEG device, can stop the data acquisition. Crazy things can happen out there in the wild. And so that's something that we have to think about dealing with. Now, I do have a question here, and it's for Luke, and it comes from Emanuele Tognoli. And she asks, and she's got a question relating to micrography, actually, and how it isolates one pathway at a time. And Westberg seemed to be working on that with a forthcoming MEG study, which should be interesting because MEG, of course, has got excellent spatial localization. Past that, we need a graded approach, dosing more or less of multiple pathways to pass out the different contribution of Meissner, Merkel, Pacini, Ruffini, and C fibers. So pulling out the different receptors, receptor types in the limb. So Luke, what do you think of that?
Yeah, that's a really great point. And it you know, especially if you're thinking of stimulation, right? Like using microneurography, not just to record the individual fiber responses, but then potentially like, you know, pump current in and stimulate. Yeah, it's absolutely a great point, because you can then start to think about, well, maybe you only want to produce the sensation of pressure, and so maybe we'll isolate a Meissner and a Merkel cell and try to stimulate specifically those afferents. But I think as we get better at that, and you know, can incorporate all types of receptors in the skin to produce very targeted effects, you know, that's maybe the goal for when we think about tactile sensation for prosthetic limbs. But with that comes, you know, it's necessary to now develop stimulation models that are able to capture those different aspects, those different fiber properties, and that, you know, that's some work that people have started to explore. So basically, you know, how do you design your stimulation paradigm so that you target these specific properties, or that the stimulation behaves in such a way that you feel mainly something like a pressure or something else.
Or you can even take that one step further, and Emmanuel's idea even one step further, where if you do what you've suggested, Luke, and you've got your known profiles of stimulation for the different types of receptors, then you can start adding the different kinds of, so you might want to have, you know, a couple of them. You might want to bring in Meissner's and Pacini, you know, receptors, and you make these complex profiles of stimulation. I mean, there's a lot of things that can be done. I think it'd be incredibly exciting in the future.
Absolutely.
So I think we've run out of time, and I would like to thank all of these three speakers for an absolutely fabulous discussion, and also fabulous presentation, very intellectually stimulating. And I love the somatosensory system. I haven't worked in it for years, and it was a great, absolutely lovely ride that you took us all on today. So thank you so much, and everybody have a rest of good Neuromatch, and we'll see you all later. Bye, everybody.
Thank you. Bye-bye.
Thank you. Bye.
Bye.